So let's finish up this unit. We may not finish it today, but this is the last section of this unit, so we will be having a test pretty soon. Probably today's Monday. We will finish Wednesday, so we're looking at probably a test next Monday. That'd be good enough for you guys. If something happens we don't get done, it'll be the next Wednesday, but we'll definitely be done by next Wednesday for sure. So look for a test either next Monday or next Wednesday, but you've had the study guide, so it shouldn't be that big of a deal. Okay. All right, hemoglobin. So let's talk about hemoglobin. So we've talked about the red cell. We've talked about the CBC. We've even talked a little bit about hemoglobin. We talked about the MCH and the MCHC. Those were dealing with hemoglobin. But let's talk about hemoglobin in general and what it is. Okay. So essentially, hemoglobin is a specialized protein that is intracellular of the RBC. It is inside the red cell, and that's where it should always stay. If it wasn't for hemoglobin, our red cells wouldn't stain. Okay, the membrane of the red cells doesn't really stain. So what we're seeing when we look on a right stain at RBCs is we're looking at the hemoglobin and how much hemoglobin is actually inside the cell. So what's the function of hemoglobin? Its main function is to transport oxygen to the lungs, or from the lungs to tissue. It also plays a role in facilitating CO2 transfer because of diffusion. It has oxygen, has lots of high oxygen percentage. So therefore, when you get into an environment that has high CO2 and low oxygen, diffusion occurs, and so it plays a role in gas exchange. Okay. I don't want you to think, and a lot of students do think this, hemoglobin doesn't, like, oxygen and CO2 don't change places with each other. Does that make sense? So like hemoglobin is carrying the oxygen molecule, but once oxygen leaves, okay, it's known as deoxyhemoglobin. The oxygen just left. And CO2 doesn't bind in that spot where oxygen left from. Okay? It does play a role to bring the CO2 in, but it doesn't really carry CO2. Okay? It carries oxygen, but it doesn't carry CO2. CO2 actually goes inside the cell membrane, or inside the cytoplasm of the red cell, and it becomes carbonic acid and a couple other things, and we'll talk about that more later on. What?
Did you say
deoxyhemoglobin. Okay. All right. Okay. So hemoglobin occupies about 33% of the volume of the RBC itself, and it accounts for about 90% of RBCs that dry weight. So like if we took red blood cells and let them dry out, most of the weight of the cell, or the mass of the cell that we have left, would be hemoglobin, because that's what makes up the majority of the red blood cell. Each cell contains between 28 and 34 picograms of hemoglobin, which is basically your MCH. When we talked about the MCH on the CBC, that parameter, 28 to 34 picograms, is the actual hemoglobin amount that's there. Okay. Remember, on our morphologies, though, we don't really look at MCH that much. Okay. We look at that MCHC for the hypochromia and normochromia and spherocytes. Okay. All right. So most hemoglobin synthesis occurs in the polychromatophilic normoblast. Okay, which is also the rubocytes. Pretty sure. Those are the two same ones. Like 75 to 80% of it is made there. Now, there is a little bit that begins at the normoblast, the pronormoblast, which is the ruboblast in my technology, or in my nomenclature. But the problem is it's not enough to detect. That's the reason we get that blue purple staining of the cytoplasm in that blast cell, in that normoblast, pronormoblast. Okay. But by the time we get to the rubocyte, or the polychromatophilic normoblast, you're starting to see some of that grayish looking cytoplasm. Okay, that's the one that's got the brain looking nucleus, you know, a little smaller. By that time, about 75 to 80% of it's made. Then the rest of it, up to 100%, is in the reticulocyte. Those residual RNA molecules and ribosomes are able to produce the rest of the hemoglobin. Okay. Hemoglobin concentration is a result of a fine balance between RBC destruction and production. Okay, and it's a very fine balance, and it has to be a certain way as you go. Okay. All right, so I want to stop right now. All right, so we talked about hemoglobin being a protein. Okay, does everybody know how proteins are formed? Like, literally, like, do you know the steps of protein formation? Because if you don't, I need to go over that with you. It'll be important when we start talking about hemoglobinopathies pretty soon.
Let's go over.
Okay, let's go over it. I agree 100%. All right, so back to biology. Okay, so in biology, what did I tell you guys the dogma of biology is? Remember the other day I wrote it on the board? I said, this is the dogma of biology. What was it? Now remember? DNA, messenger RNA makes what? Say it. Protein. Okay? That's the dogma of biology. In other words, that's the whole complete theory of how biology works. Okay? So, we just said that hemoglobin is a protein. So we know that we've got a cell that has a nucleus. And we know that in our case, all this is always happening in the normoblast. Okay? The pronormoblast, right? Okay? Inside this pronormoblast. Alright? So, we know that it's a complete nucleus. So we can do transcription, we can do translation, we can do whatever we need to. Mitosis, whatever needs to happen, because we have a complete nucleus. Okay? So, my problem is, if I magnify my cell up really, really large, and we look at all the other organelles and different things that are in the cell itself, we'll find part of the ER, endoplasmic reticulum, has these dots all over it. Okay? And then inside our cytoplasm, we have these dots all over the place. Alright? What are these dots? Those are ribosomes. Ribosomes. Okay? So if we go back to biology, we know that ribosomes are where protein synthesis occurs. Okay? That's where it takes place. That's how we make a protein. The problem that we have is our code's in here. Right? So for us to make a protein out here, we have to get the code from here to here. Now DNA doesn't like to leave the nucleus. Okay? Matter of fact, it never leaves the nucleus. Okay? The only time that DNA doesn't have a nucleus is when it's doing cell reproduction. And it's during the cell cycle, right? Through mitosis. And then the nucleus dissolves, right? Okay? But other than that, the DNA always stays in the nucleus. So how do I get my message or code to my ribosome from my DNA? Messenger RNA. That's exactly right. So what I do basically, or what happens, is we know that DNA is a double helix, right? Okay? We know that DNA is basically a bunch of A's, G, C, and T, right? Now, A is adenine, G is guanine, C is cytosine, and T is thymine. Okay? These are our nitrogenous bases. These are our codes for making proteins. Okay? Based off of their binding. Okay? We know that G and C are always together, and A and T are always together. These are known as purines. Okay? A and G are purines. And T and C are pyrimidines. Okay? And that's important to you because these guys are going to come into play later on when we start talking about uric acid. Later on in chemistry, clinical chemistry, we'll discuss an analyte called uric acid. Uric acid is a byproduct of the breakdown of purines. Okay, so the adenine and the guanine that you guys will talk about here. Okay, so basically you have, wherever you have an A, you're going to have a T. Okay, and there's a double bond between them, and G and C has a triple bond. I believe that's right. Maybe wrong. Maybe right the opposite. I haven't looked at it lately. But either way, one has a double bond, one has a triple bond. Okay? So, basically what happens when we get ready to make a protein, and of course this is occurring all the time in the, when we talk about hemoglobin, we're talking about this particular protein is always occurring in those erythroblasts. Okay? Up to a certain point. Okay? After they lose the nucleus, for just a little while until it becomes mature. Once it's mature, this stops. It's never happening again. Whatever hemoglobin's in the red cell, that's all it's ever going to have. Alright? Okay? So what happens in our body I'm just making crap up at this point, okay? Alright, so we have a shape, okay? What's going to happen is we're going to have a polymerase, okay? You guys you're blessed enough to come into a time in the health field where PCR stands for polymerase chain reaction. Is common thing. You're going to laugh at me, but when I was in MLT school, we didn't even know what the crap that was. I graduated in 1996. Okay? It doesn't sound like that too long ago, but it just shows you how fast technology is taking us, okay, forward. But I didn't even know what PCR was until I started in 2002. So from 96 to 2002, PCR became a big thing. And when I got to JSU to start my bachelor's degree to become a teacher, my high school teacher, in my genetics class, we did PCR things. We did a transformation. Y'all ever heard of that? Transformation lab. It's basically when you take a gene from one organism and put it in another one. So we did that. We took a gene, actually we took several genes. They're called plasmids. Ms. Cole probably taught you guys about plasmids, right? Plasmids, we know bacteria don't really have genes. They have plasmids and then that single strand of DNA, okay? So you can take a plasmid from something else and you can insert it into another organism. We created an E. coli bacteria where we took a plasmid from a jellyfish, and then we took a plasmid from an organism that was ampicillin resistant, and we put them in that one E. coli organism, and we had E. coli that glowed, and then it was also resistant to ampicillin. Great. So it's pretty cool, you know. But anyway, so but we do have this enzyme polymerase. So polymerase basically comes in, and then you have another enzyme that's called helicase. Helocase, I think. Okay. Maybe helocase. I can't remember if that's an A or an O. Okay. But essentially, what this guy does, it unzips it. It basically breaks the bonds of those hydrogen bonds. Okay. It basically breaks the helical shape of it. Okay. So what it does is now we have this. Right? And these are A's, T's, G's, C, C, G, A, T. Okay? So that means we have A, T, C, G, G, C, A, and T on this side. Okay? So basically what we did, we just unzipped it. Alright, so let's say that this gene is what we need to make our hemoglobin protein. Okay, the globin part. Okay, let's go ahead and make that clear real quick. Alright, hemoglobin. Okay, there is one heme molecule and two globin chains. Okay, in hemoglobin. Alright, and one. Alright, there's actually, hemoglobin is kind of shaped like this. Now, there's a picture in your book in just a minute. Okay? And you have salt bonds that hold these four together. It's a tetramer. Okay? And then, like right here inside, there is a chemical structure known as heme. Okay? Alright, so that's heme. And then this is the globin chain. Okay? So technically you have 4 and 4. Okay? But there's only one heme on each globin chain. Okay? And I've got pictures of this on the PowerPoint. I'm just throwing it up there so you know what we're talking about. Okay? But what we're making is this portion. Okay? Heme itself is not a protein. Okay? Heme itself is actually a chemical. It's actually a pyrrole something ring is what it's called. Okay? Chemically. Okay? But the globin chain is a protein, and that's actually the most important parts of hemoglobin, is that globin chain. Okay? Alright, so globin, to make the globin chains, has to take place this way. So what we have, basically when our body says, okay, we need to make hemoglobin. So it unzips our DNA using helocase. Okay? And then what's really cool, and we don't talk about this that often, we're going to start basically chemically summoning these nitrogenous bases. Okay? So, we're making DNA. This is DNA, but we're making RNA. RNA has A, G, C, and then instead of having T, it has a U. And the U is called uracil. Okay? Uracil. Okay? Uracil. Alright? So everywhere you're going to have an A, instead of having a T, you're going to have a U. Does that make sense? Okay? So when we're making RNA, T is replaced with U. Okay? So, but U and A always bind because that's how it works. Alright? So U, now this is a T, so it has to bind with an A. Alright? This is a G, so there has to be a C, a G, G, C, U, and then we're going to say A. And we're going to make this a short piece. Now these things can go on for 1,000 base pairs normally. Okay? Just depends on what's going on. Alright? So now our body gets to a certain spot and it says, okay, that's what I need. That's my code. Okay? So I'm done. So what happens now, this breaks loose. Alright, so what's cool? Okay? In our body, we have an enzyme, a protein, that circles the process of making this messenger RNA. All right? So as it's making it, it's going up and it is checking it. It's checking it to make sure that we're getting the right things in place. If something happens, and let's say a C gets there with a T, it stops and it says, that's not right. And it cuts it out like little scissors. They're called restriction enzymes. And it'll come in and it'll cut it out and it'll summon the right thing. That's the reason mutations in DNA are very, very rare. Okay? Usually if you have some kind of a mutation, it's because of some outward thing has caused the mutation. It's usually not happening because of just natural replication. Does that make sense? And the reason it is because we have that cool protein that sits there and checks it as it goes. Okay? That's the reason most of the time you don't see a mutation from one cell, then goes through mitosis, and it forms another cell. Okay? And that cell's mutated. It just doesn't happen because of this. Okay? Alright, so then what happens is, alright, so let's stop right here and let me give you a new term. Okay? This is called transcription. Okay? Very important term in biology. Okay? Transcription is essentially DNA making messenger RNA. Okay? That's transcription. Alright. So, you guys ever been to court? Or been to a courthouse? Watched the show with the courthouse? Got a little woman or a little man standing up there doing this the whole time everybody's talking? You know what they're called? Transcriptionists or transcribers, right? Because they are making a transcription of the court proceedings. Okay? Let me ask you this, Haley. You the baby in the class. Would you be able to walk up and take that lady's paper and read to the judge what she just wrote? You would not. Because her little machine actually is like shorthand. You know they used to teach shorthand in college? If you was ever going to be in business or whatever, that was actually a required class, was shorthand. And it was like little symbols and crap, you know? But anyway, so that's what her thing does, okay? So transcription, she made transcription. Well, that's what your body does. It's making transcription. It's saying, I got to make messenger RNA, all right? But at this point, we can't read the messenger RNA, all right? So it's got to go out of the nucleus. And how does it do that? Because our nucleus, if you remember from biology, has pores, right? Y'all remember nuclear pores, where y'all taught that in biology? Your nucleus is not solid, it's like, it's got holes in it all the way around. Okay, those pores are so messenger RNA can get out. They do other things too, like the nucleolus makes ribosomes, so they have to get out and those kind of things, all right? So once we have our code, then we need to go to a ribosome. All right, so I'm going to erase this part right here. Okay, so I'm going to magnify up a ribosome. Ribosomes are kind of unique shaped. They are actually have what's called a small subunit and a large subunit. I know, very creative, right? Okay? And inside these things, they have a P site and an A site. Okay? Now, at this P site and A site, they have sensors, basically, is what they are, between the two. Okay? Kind of like your Walmart reader that's reading your barcode. Okay? Hey. Reading your barcode as you slide your stuff. Okay? So what this guy's doing, though, is we're reading messenger RNA. So we've got our strand of messenger RNA. All right, here, this is our messenger RNA. Okay, so what's it reading? Okay, it is actually reading every third pair. Okay, these third pairs, each one of these guys is known as a codon. Okay, a codon is basically the code for one amino acid. Okay, you know that's the building blocks of proteins, right? Building blocks of proteins are amino acids. I used to tell my high school kids, don't go home and call your mama amino acid. You get in trouble. So, amino acids. Amino acids, a codon codes for it, okay? There are 24 natural amino acids. I'm just going to put AA, okay? Anytime you see abbreviation AA, means amino acid, okay? 24 natural amino acids. Now, there are several codes that can code for the exact same amino acid, okay? So later on when we get in, let's see where we do that, clinical chemistry, there's actually a section in your book that has amino acid box, and it kind of tells you what they are. We start talking about molecular things in clinical chemistry. We'll talk about this again, okay? So, this guy enters the A site, and so it is going to read that first codon, okay, whatever it is, all right? It's going to say, okay, that one is, let's say, glycine. Glycine, maybe it's C, okay? Can't remember, I think it's C, yeah, glycine, okay? Glycine, all right, so it says, okay, chemically I know I need a glycine, all right? So how do amino acids get to the ribosome? They just kind of floating around in our body, hanging out, going through our bloodstream, saying, hey, need to be used. Are they doing that? No. Where are they at? Okay, so let me go ahead and give you guys a secret. All right? In lab school, in general, if I ever say, where is so-and-so located? Where is so-and-so broken down at? Okay? 9 times out of 10, it is going to be liver. Okay? So, you can be smart, and then every time I say something, say liver, and you have about a 95% chance of getting it right. Because that's where everything occurs, just about. When destruction and storage, it occurs in the liver. Okay? Okay? Alright, so, let's test the theory. Where are amino acids at? Liver. Yes, they are in our liver, actually. We have these pools of amino acids that basically your liver plays a role in breaking down hemoglobin. It plays a role in breaking down, heme, and then it breaks down globin chains also. And then those amino acids are basically recycled and stored in the liver pool. Okay? So, how do they get from the liver to the ribosome?
Liver.
Bummer.
What's that? And it's shaped just like this. I tell you what, if you can tell me what this is, we'll just let you skip the rest of this little section. What is that? Alright, what if I did this? What if I put
RNA messenger.
well, it's definitely RNA, right? Okay. So what kind of RNA is it? Transfer RNA. This is the shape of transfer RNA, okay? The amino acid is always hung here on the transfer RNA. It's basically a transport system, transfer RNA, okay? But on the transfer RNA, this part right here is the most important part, okay? It actually has 3 nitrogenous bases there that are called anticodons, okay? This guy is always going to be the opposite of this guy, whatever the codon was that it read, okay? So like for instance, if this was our codon, okay, let's say UAG was our codon, the anticodon would be AUC, okay? And so what's going to happen at this A site, it's going to read that codon, and it's going to say, okay, UAG, I need anticodon AUC to bring me the amino acid glycine. Okay? Does that make sense? So then it reads it, that other codon comes in, the little amino acid is stuck to the end of it, of course. Alright? So it says, okay, I got it. That's the one I need. So it shifts over, and basically it attaches the amino acid. Okay? This process happens all the way till it reaches a stop codon. One of the most common stop codons is AUG. Okay? AUG. This is known as a stop codon. Alright? So what's going to happen is somewhere at the end of this messenger RNA is going to be a AUG. Okay? So once it runs through, and as it runs through, this amino acid chain is going to get larger and larger and larger until it reaches that stop codon. Alright, when it gets to that point, your ribosome says, okay, now we're done with it. We're done. Alright? So the messenger RNA gets basically recycled. Those A's, G's, U's, C's, and T's break down. They go back and they're kind of hanging out in the cytoplasm of your cell. Okay? So we make it, now we've got our protein. Alright? Polypeptide chain. Okay? It's called a polypeptide chain. Alright? These little bonds are known as peptide bonds. Okay? They're what holds amino acids together. Okay? Any questions? Alright? So, this process is called translation. Okay? So we took the transcription, our messenger RNA, and we translated it into a protein. Okay? Now, this little protein here is known as the primary form. Okay? It has no function at this point. Okay? So, think back to your biology. All right? Where does a protein or a primary form of a protein have to go now? It has to go to the endoplasmic reticulum. So then, once it was released from the ribosome, it's transported to the endoplasmic reticulum. There it is modified and adjusted to be able to be kind of like this now, okay, which is known as its secondary form. Okay? Then once it gets to its secondary form, through enzymatic reactions in the ER, then it goes to its tertiary form. Okay? Once it is in its tertiary form, you see more binding together. And so it gets condensed. okay, like this. All right. Now, at this point, it is functional, but it's not fully functional, okay? It doesn't become fully functional until it gets to its quaternary stage, okay? And when it gets to the quaternary stage, that is what this guy is, okay? So not only is it fully functioned, but it's bound to other proteins to become a functioning protein for the enzyme or hemoglobin or whatever is necessary. And it's known as quaternary. And that's on one of the things. We'll get there in just a minute. I think it's right here, isn't it? Okay? And it's on the PowerPoint slide too. Pretty sure. Okay? All right. Questions about transcription, translation. I would know this if I were you. Okay? Hint, hint. Okay. All right. So let's go on. So normal concentration adult males is about 15 grams per deciliter. Females tend to be a little bit lower. About 13 grams is what females tend to be. And that's assuming that they have 5 liters of total blood volume, okay, which is what most males have, okay. Normal RBC lifespan is 120 days. We've talked about that. So basically 1/120th, which would be about 1%, right, of the total amount of hemoglobin is lost daily. So that's because that's how many red cells we lose daily. So for us to do this, when we lose those red cells, remember I just said a little while ago that hemoglobin production is a delicate balance between destruction and production of RBCs, okay? So if I'm losing 1% of my red cells per day, I have to make at least about 1% hemoglobin to refill it, okay? So that's equivalent to about 6.25 grams a day, okay? Which makes up about 750 grams of your total body mass of hemoglobin, okay? Which is 30 picograms for your MCH. And that should make about 10, is that 100, what is that, 100 million? 11? So let's see, 9 is a billion, right? So 10 would be 10 billion, 11 would be 100 billion, right? So about 200 billion red cells per day is what we have to make to maintain our balance, okay? All right, so talk about the structure of hemoglobin. Now that we've talked about proteins and what they do, all right? So proteins or hemoglobins are 4 globular subunits. These are what's important to us, those globin chains, because they're the ones that's actually going to determine the function and identity of your hemoglobin. Okay, and I know that sounds crazy, but trust me on this. There's actually, I think, 1,500 different variants of hemoglobin that we know of today. All right, let me ask you this. What's a variant? You guys we're living in the world of COVID, and every other week we have what? A new variant, right? So what is a variant?
Another form.
It's another form. That's exactly what it is, another form. So we know that there's at least 1,500 different variants of hemoglobin. HGB is how they abbreviate hemoglobin, okay? So that just kind of lets you know that. But we want hemoglobin to be a certain way, all right? We have a globin chain that right in the center of it, I wish I had some like colored chalk. I know colored chalk. I want to buy some colored chalk, okay? Right in the center of the globin chain is a heme molecule. We're going to talk about that in just a second, okay? That's the heme, and then this is the actual globin chain. All right? Now, hemoglobin is a tetrad. Okay? Tetrad, I think is how it's spelled. Okay? What a tetrad is essentially just like what it sounds. It's 4 of the same things bonded together. Okay, in our case though, it's unique bonds. Okay? There are 2 alpha and 2 beta. Okay, now that doesn't make sense to you right yet, but it will in just a minute. Okay? 2 alpha and 2 beta give us hemoglobin A1. Alright, we refer to it as A1 because there's actually a different form called A2. Okay? Sometimes you may just see it as A, hemoglobin A. Alright? But there is a hemoglobin A1. Alright? As we go. This guy should make up about 97% of the hemoglobin inside a red cell. Okay? This is our normal hemoglobin. Okay? But we actually have genes in our body that make lots of different globin chains because there are certain combinations that are necessary from the time we're conceived to the time we die. Okay? Or at least become an adult. Alright? So we're going to talk about that in just a minute. Alright? So heme is a tetrapyrrole ring. Alright? We're going to talk about that in just a minute. With a ferrous. Okay? This is very, very important that you know ferrous and ferric. Alright? So if you guys have taken chemistry, this of course is the Latin term, right? This is ferrous. This is basically iron 2. This is ferric. This is iron 3. Okay? Alright. Here's the problem. Okay? Most of the iron that we take into our body, alright, when I eat steak, liver, whatever I eat that puts iron into my body, it comes in as the iron 3 steak. Okay? So we have to have an enzyme, a protein enzyme, that converts it. Alright? An enzyme that converts it to iron 2 so that it can be used in that heme ring there. Okay? Alright? The reason it does is because this guy, iron 3, doesn't like oxygen. Okay? It actually will not bind to oxygen like it needs to. Okay? So we have to have it in this state for it to bind with oxygen, iron 2. Okay? We're going to talk about an iron 3. We actually have a form of hemoglobin that is bound to iron 3 at one part of the production, or the breakdown actually. Okay? Questions about hemoglobin so far? Okay. So, each heme structure, each heme subunit can carry 1 molecule of oxygen bound to the central ferrous iron. So that means that each hemoglobin molecule, okay, in your body, with the 4 globin chains, carries 4 oxygen. Okay? So what you have is you have a subunit here. So I'm just going to put right here. So this is 1 hemoglobin molecule, carries 4 oxygens. Alright? Now later on we're going to talk about this thing called saturation and unsaturation and all that. Okay? Saturation means that there's 4 oxygen molecules. Unsaturated means there's some free iron. In other words, it doesn't have an oxygen molecule bound to it. Alright? Okay, let's talk about globin chains. This is important. So this is what we were talking about before. So you have 2 chromosomes. Okay? So how many chromosomes do we have in our body? How many chromosomes do we have in our body as an adult? Okay, so
23.
if I got my normal blast, and I look inside there, how many chromosomes are inside that DNA, or in that nucleus? All right, look, people, y'all are like advanced biology here. Okay, this is considered advanced biology. You need to know this if you don't know this. Okay, we have 46 chromosomes in our cells. Okay, with the exception of what 2 cells? Sperm, eggs. If you have eggs, you're a female, you make eggs. Okay? If you have 2 X chromosomes, you make eggs. There's no freaking way around it. That's what you do. Those have 23. If you're a male, you are XY, you produce sperm. Guess what? It has 23. When those two get together, 23 plus 23 makes what? 46. So from here on out, if I say, how many chromosomes are in a human, what are you going to tell me? And liver's not the answer this time.
Liver.
46. Okay? You need to know that. That's basic biology, guys. I mean, I promise. Okay? All right. So, back to that. All right? So, a lot of times you'll hear people say you have 23 pairs of chromosomes. Okay? So that may be where y'all are getting the 23 from. I don't know. Okay? But 23 pairs still means how many? 46, right? Okay? So when you have 23 pairs, that means you have 2 of each chromosome. Okay? 1 through 23. Right? Okay. In our body, we have globin genes. Okay, so that means genes that make globin change. There are 2 chromosomes. Chromosome 11 and chromosome 16. Okay? Chromosome 11 and chromosome 16. All right, so what does that actually mean? That means I have 2 11s and I have 2 16s if I'm normal, right? This is yes, this is no. That's normal, right? I should have 2 pairs of each one. Okay. Located on these 2 genes are alpha and non-alpha. Okay? All right? Trying to get to that. We talked about that already. Okay, here's a picture. Okay? All right. Primary, secondary, tertiary, quaternary. Talked about that a while ago, right? So primary is just the chain. Secondary is starting to kind of be arranged the way it needs to be. All this is happening in the endoplasmic reticulum. Then, of course, it begins folding in on itself. Basically, at the tertiary, it is functional, but it's not completely functional. Then by the quaternary, it forms bonds with other proteins to form what we need, okay, and other ligands, all right? So this is a different picture also. Kind of shows you what's happening. All right? So it's going to talk about heme first. So I tell you what, let's just go ahead and talk about these right here, just this part, and then we'll talk about this part in just a minute, okay? Later on, it's on in there. But it gives us some understanding, okay? So you have alpha-like genes. They're known as alpha-like, but they are actually alpha, which is abbreviated with a little a, and they are zeta. All right, zeta has a kind of a weird, like Z looking deal there. Okay, zeta, alpha and zeta. Okay, now alpha is very important to us. Okay, because it is found in all adult hemoglobins. Okay, when the baby is in the embryonic phase, so remember back to hematopoiesis, we talked about the yolk sac first, and then the liver began doing hematopoiesis. At that point, it's actually a different type of hemoglobin. And we'll talk about it in just a minute. There's actually 3 of them, Galler-1, Galler-2, and Portland. Okay? This zeta plays a role in those embryonic ones. Okay? So like adults would never have zeta in their hemoglobin. Okay? But alpha is always there. Alpha is in every one of our adult hemoglobins, A1, A2, and even in fetal hemoglobin. Even though we don't have much fetal hemoglobin, we still have a little bit even as an adult. Okay? All right? Non-alpha, though, there are 1, 2, 3, 4 of these. Okay? They're just known as non-alpha. And they are epsilon, which is an E, like a little e, or a big E, I'm sorry. Delta. Delta is, it's always a weird symbol too. Yeah, it's like a
Very curvy B.
yeah, like that kind of weird looking.
Yeah.
Okay, you got it up there. Y'all can draw better than I can. Beta, which is the one that is very important to us, because this is what makes our adult hemoglobin, right? We said 2 alpha, 2 beta. All right, so that is very important to us. And then the other one is gamma. Now, gamma is unique because there's actually a gamma A and a gamma G. And we'll talk about those later on. 6, 7. So there's actually a total of 7 globulin chains that are available, okay? And then gamma is that little weird Y shape, isn't it? Yeah, it's like a Y, a little Y shape, okay? Now gamma becomes important to us. We'll talk about this in just a little bit. But gamma and alpha are the 2 that makes fetal hemoglobin. So once about 12 weeks into gestation, the baby converts from that Portland and Galler to fetal hemoglobin, and then they make it the rest of their embryonic and fetal stage, and then even for a little while after they're born, for a few weeks after they're born, and we'll talk about that later on. Okay. In a minute, though, I'm going to tell you which genes they show up on, and that's where we'll get into those chromosomes in just a minute. Okay. So let's talk about heme first. All right. So heme in general, okay? All right? So 2 identical alpha-like, 2 identical non-alpha, okay? So every type of hemoglobin, no matter what it is, whether it's at that fetal embryonic stage or as an adult, you're going to have 2 alpha-like and 2 non-alpha, okay? No matter what they are, all right? So one's going to be on one chromosome, one's going to be on the other, okay? All right, so 4 subunits of hemoglobin, each consists of a heme group surrounded by a globin chain. We've talked about that, held by salt bonds, hydrophobic contacts, and hydrogen bonds. Okay, so I do want to mention this. We'll talk about it a little bit more, but these salt bonds and hydrophobic contacts are very, very important, okay? Because, okay, we're going to talk about this thing we called tense state and relaxed state. Okay? And what that essentially means is your hemoglobin in a tense state is like squeezed down, holding that oxygen molecule in place in the heme group. Okay? In the relaxed state, it opens it up to be released. Okay? These salt bonds and hydrophobic contacts play a huge role in that tension and relaxation of the hemoglobin. Okay? Now, it's not important right now, but a little later on it will be when we start talking about hemoglobin oxygen curve. All right, we're going to cut the heat off, guys. Reach better cut that heat off. If that one's on.
Yes.
Somebody might cut them on. Man, cut the fans on.
It's not.
Huh? Is there something on? One of them's on back there, isn't it? What's that I hear roaring?
That's the AC over there.
Oh, is it the projector?
No, the AC over there.
The AC's on.
I don't know. Heat or something.
Either way, it's warm. I'm sweating. Might be a clench in church. We're going to open the window. All right, it's the projector that's actually making the noise that I'm hearing buzzing. I thought it was the heaters because it just kept getting hotter and hotter in here. All right, so tetrahedral formation, that's what we've got with those four globin chains as we go, like what we talked about here, alpha 1, alpha 2, okay? And then the beta 1 and beta 2. This is normal, okay? So right here is the heme group that's inside there. And then inside here is where your iron 2 would be bound with oxygen, okay? So you're going to get that kind of rust. That's essentially what it is. It's iron oxide, okay? Inside. That's where you get that metallic taste in your mouth when you get your blood in your mouth, okay? As you go, all right? Okay, all right, so let's talk about what time is it? How are we doing? We're doing good, okay? 55 minutes, we're in good shape, okay? So, heme. So let's talk about heme in general. All right, heme is iron chelated, okay? So what does chelate mean? What's chelate mean, Caleb? You usually know all this stuff. What's chelate mean?
Okay, so I went over the chapter.
Hmm?
And I had it written down and it's in the books over here.
Okay.
Somewhere.
What's chelate mean though? You got
But
it.
to chelate something means to form a chelate with it.
Which means what? To what? Bond. It's to grab it, okay? So you remember in phlebotomy, we talked about how anticoagulants work. We talked about EDTA, and I use this term chelate calcium. That's what it does. So when EDTA comes in contact with calcium in the blood, it grabs it and forms a bond with it. That's the reason it works, because calcium is necessary for clotting. So if calcium's not present, your blood can't clot. Okay? So in our body, heme, the heme molecule, at this point, we're not talking about hemoglobin, we're talking about heme itself, chelates iron. Okay? And it has to chelate it in that ferrous state for it to be normal hemoglobin. Okay? So porphyrin. Porphyrin ring, it forms what's called a porphyrin ring. Protoporphyrin 9. Okay? So later on, a little bit further into the semester, or actually in the next unit, we're going to talk about some things that are abnormal with hemoglobin. Okay? One of the things one disorder that has to do with heme is known as porphyria. Okay? You guys ever heard of porphyria? No. Okay. I think that's how it's spelled. Porphyrias. Okay. Porphyria is a genetic abnormality in the production of protoporphyrin rings. Okay. So history will tell us about vampires. Right? They believe that they're really vampires. Photosensitive, you know, if they go out in the light, they turn to dust, right? In the moon, sun, they go out in the moon. Which never makes sense to me, you know, scientifically. Moon is sunlight, right? Moonlight, sunlight, same thing, right? Should have died at night too, shouldn't they? If they went out in the moonlight, okay? Sunlight, okay? Fine hair on them. They actually, vampires and werewolves all came from porphyrias, really and truly, okay? Bloody teeth, okay? So porphyria, porphyrin, which is the compound here, porphyrin, these people that have porphyria, they accumulate porphyrin in their teeth and in their urine. So their urine will glow red, okay? And actually, when it comes out, it looks like port wine, what we call port wine. So it's like a reddish purple color, okay? And then if you put a woods lamp or a UV light in their face, their teeth glow red. So therefore, blood on their teeth, vampire, right? So all of that stuff comes from a disorder called porphyria. These people were genetically, okay? So you remember, I told you guys, I think I mentioned this in phlebotomy when we were talking about it 131. The royal families in Europe, you know, they were all inbred. There was all cousins, married cousins, and all that kind of stuff. Well, porphyria is a genetically disorder. And so you start seeing an increase in these. So there was several of those princes that they had over there that they thought were vampires. And all it was is they really had porphyria. Okay? It's a disorder. All right. So we'll talk more about that later on. All right. So heme, that protoporphyrin ring, is composed of a flat tetrapyrrole ring, and I'll show you what this looks like, and then it has a ferrous iron in the center of it. Okay? And then this iron must be in the ferrous state for oxygen to bind to it. If it doesn't, it will not bind to iron 3. Okay? All this begins in the mitochondria. This is the important part I want you to know. Okay? So if I ask you on test, how does heme production begin? You need to know this. Condensation of glycine and succinyl-CoA. Okay? Succinyl coenzyme A, also known as CoA. Succinyl-CoA. All right? These guys, through enzymatic, what? No worries.
What was your question?
I said if I ask you how heme production begins, okay, you say glycine, succinyl coenzyme A. Okay? These two, through enzymatic reaction, form 5-aminol vulinic acid, ALA. Okay? That's all I want you to know. That's all I really care about right there. Okay? For heme production. The beginning of it, and then the last thing I want you to know is do know that this is the limiting step. Okay? Rate limited. You know what a limiting step is? Have y'all ever heard that in chemistry? Huh? When you guys took chemistry, did y'all talk about limiting steps? Okay, so all right. If I have stars and circles, okay, okay, and I have to put a star and circle together to form a compound, which one of these is my limiting factor?
Stars.
My circles, right? Why my circles?
Because there's not as many.
Because I have less of those, right? So at some point, this one bound this one, this one, this one, this one, this one, and this one, this one, and this one, this one have extra stars. Does that make sense? So my limiting reagent was this one. Does that make sense? That's what these guys are, okay? These are limiting reagents, or limiting, step limiting reactants. So in other words, if they're not present, you can only make so much hemoglobin. Does that make sense? So these guys are very important. And notice what each one of these are, with the exception of pyroxyl phosphate. ACE and ACE. What I tell you guys, anytime you see ACE, it means what? Enzyme. So enzymes are so important to us. And without them being present, you can't go any further in the steps. Okay? And that'll come into very important play next semester when we talk about, in your analysis, we talk about IEMs, inborn errors of metabolism. It's usually genetic disorders where they don't make a particular enzyme. And so that enzyme missing limits their production or breakdown of certain products in their body. Okay? So that comes into lots of play to us. Okay? All right? Also, iron is considered a limiting reagent also. You can only make hemoglobin if you have iron. Our body's not, I taught you guys, our body's really efficient. Okay? It's not going to make stuff if it doesn't have all the necessary products to make it. Okay? So iron would also be one. All right? So basically what happens is we get coproporphyrin, porphyrinogen, okay? So look, this step here, all of this stuff starts in the mitochondria, okay? Producing ALA, okay, with the enzyme 5-aminolevulinate synthase, okay? Then the iron, okay? Now iron's not there, okay, but it also acts as a limitant. We hadn't got to iron yet, production, okay? Then that part of the ALA moves to the cytoplasm of the cell, okay? ALA moves to the cytoplasm. Then, through enzymatic reactions, okay, all right, so coproporphyrinogen re-enters the mitochondria to form our protoporphyrin ring 9, okay? So that's another thing I want you to know about heme production. Know that how it begins and the end product is a protoporphyrin ring 9, okay? I don't really care that much about the steps in between. You guys, eventually I pray and hope that you guys will continue on and become MLSs, okay? Go to MLS school somewhere, get your bachelor's degree. Then you can talk about in between, okay? But I want you to know the beginning and the end product, which is this protoporphyrin 9 ring, okay? So then once this is formed in the mitochondria, this goes back into the mitochondria, this part is happening in the cytoplasm. Then it enters the mitochondria, and then through an enzymatic reaction with ferrochelatase, that protoporphyrin ring binds with iron to form heme in general, okay? Now in your book on page 81 in your book, figure 6.3 shows all the detail of this formation. What part occurs in the mitochondria, what part forms in the cytoplasm. Okay? All right, so the enzyme that's important, ferrochelatase. Ferrochelatase. Without it, iron can't bind to that protoporphyrin 9 ring. Okay? So it has to be there to form the complete heme molecule. Then, of course, heme leaves the mitochondria and combines with a globin chain in the cytoplasm. Okay? Questions about that, guys? Okay. All right, there it is, figure 6.3. So succinyl-CoA plus glycine, okay, with ALAS2, which is the synthase enzyme, okay, forms ALA, which is that 5-aminolevulinate acid. Okay, so then once this is formed, we go to the cytoplasm with the help of another enzyme to get it across from the mitochondria into the cytoplasm. Okay, then we get to the porphyrinogen down here through some more enzymatic reactions. So eventually we get to coproporphyrinogen 3 and 1. Okay, 3 goes back into the mitochondria. Then it is converted with another enzyme to your protoporphyrin 9 ring. Then through ferrochelatase enzyme, iron binds with the 2, and that forms our heme molecule. And then the heme leaves the mitochondria, and it goes and binds with a globin chain. Okay? All right? That's enough for the day. All right, so we'll pick up on the globin synthesis Wednesday. Okay? So chromosome 11 and 16 is where we'll start talking about those. Okay?